This is Chapter 12, Part A on the central nervous system. So the central nervous system consists of the brain and the spinal cord, so kind of the central portion of the body. So throughout our evolutionary history, a process called cephalization occurred. So this basically means that we had evolutionary development of the anterior or the front part of the brain. So this resulted in an increase in the number of neurons and brain cells. And we see the highest levels of these reached in the human brains, which helps account for our remarkable intelligence. So this is showing kind of the progression of brain complexity through the species. So fish have the simplest, most rudimentary type of brains. Amphibians, reptiles, we see some growth, whereas birds, like a goose here, is much larger. So birds are fairly intelligent in the animal kingdom. And then here's a mammal or a horse. So embryologically, the brain and spinal cord begin as a neural tube. So pregnant women are encouraged to take their prenatal vitamins so they get plenty of folic acid because it's important to help prevent these neural tube defects. So at the very beginning stage of development, we have this tube. So if there are any defects or holes in the tube, those effects will be felt kind of throughout the development of the brain. So the neural tube's anterior end expands, and these constrictions, these different regions, are going to form the primary vesicles. So we have three primary vesicles: the proencephalon, or the forebrain; the mesencephalon, which forms the midbrain or the middle portion; and the rhombencephalon, forming the hindbrain or the back posterior portion. So the very posterior end at the very base of this neural tube will go on to become the spinal cord. So these three primary vesicles will then develop and give rise to 5 secondary brain vesicles. So this forebrain divides into the telencephalon and the diencephalon. The midbrain's pretty much going to remain the same, undivided. It's still the mesencephalon here. And the hindbrain becomes the metencephalon and the myelencephalon. Okay, so 3 primary brain vesicles dividing into 5 secondary brain vesicles. So then the development of these vesicles into adult brain structures, this telencephalon gives rise to the cerebral hemispheres. So both of these together on either side of the brain make up the cerebrum. So we have the cortex, the white matter, and all of the basal nuclei. The diencephalon will develop into the epithalamus, hypothalamus, and the thalamus, as well as the retina in the eye. Mesencephalon remains the midbrain, the midbrain portion of the brain stem. The metencephalon then becomes the pons and the cerebellum. Myelencephalon develops finally into the brainstem portion with the medulla oblongata, and then that final central cavity becomes the spinal cords and the ventricles. So as the brain develops, it's actually growing faster than the skull. So in order to kind of fit in that limited skull space, it has to fold in on itself. So by week 5, the forebrain has kind of folded down toward the brainstem. And then finally, around week 13, the cerebral hemispheres grow and double back over that brainstem and the diencephalon, while creasing and folding to increase its surface area. So this is why we see all of these ridges and folds on the surface of the brain tissue. So adult brains have 4 regions. So the cerebral hemispheres are what we see on the outer structure of the brain, all these folds and ridges on the surface. The diencephalon, shown here in purple, is kind of inside the brain. The brain stem, shown here in green, consists of 3 regions: the midbrain, the pons, and the medulla. So remember we said the midbrain pretty much remains unchanged, that mesencephalon, throughout the development early on. And then finally, the cerebellum. The cerebellum is this kind of low occipital region lobe at the very base backside of the brain. So, looking at the gray and white matter in the central nervous system, we said before in last chapter, if it's considered gray matter, that means that it has primarily non-myelinated neurons, whereas the white matter does contain the myelin. So, remember we said myelin was kind of a fatty type protein substance that encases and insulates the neurons. So it's kind of like marbling on a steak. So the white matter is kind of the marbling of the central nervous system, showing us that that fatty myelin is present. So the basic pattern of this gray to white matter found in the central nervous system is we have a central cavity of gray matter kind of in the core of the tissue with white matter on the external portion. So the brainstem also has some additional gray matter nuclei kind of scattered throughout the white matter, similar to what we see in the cerebral hemispheres or the cerebrum. So we have a cortex of gray matter on the outer layer. So this is kind of where your higher mental functions and thinking takes place. The cerebrum and cerebellum also have a few scattered regions of gray matter nuclei within the sea of white matter. And so we said the ventricles were kind of the chambers left over from development of that neural tube. So these chambers are filled with fluid and continuous throughout the central nervous system, the brain, and the spinal cord. These chambers or ventricles are filled with cerebrospinal fluid. And remember from last chapter, when we talked about neuroglia, we said that the ependymal cells were the neuroglial cells that line these ventricles. Right? So they have the cilia or these little hairs that help to just keep this fluid, cerebrospinal fluid, circulated throughout the central nervous system. So the ventricular system contains these large C-shaped ventricles on either side in each hemisphere, and then they're going to be connected by the third ventricle kind of in the center of the brain, and then leading out into the fourth ventricle down into the spinal cord. So the lateral ventricles are connected to this third ventricle via the interventricular foramen. So again, interventricular just means between the ventricles, and remember from the bones, foramen means hole. So this is a hole in between the ventricles that's going to connect them. This third ventricle, as we said, kind of lies deep within the brain, within the diencephalon. So a lateral view, you can kind of see it outlines the shape of that diencephalon structure. Third ventricle is connected to the fourth ventricle here through the cerebral aqueduct. So it's just kind of a flow channel for that cerebrospinal fluid, and it's going to be continuous with the central canal of the spinal cord. The cerebral hemispheres are going to form the superior part of the brain and about 83% of the actual brain mass. So majority of your active brain is going to be the cerebral hemispheres. So surface markings, looking at the surface of the brain. So a gyri or a gyrus are the large ridges, kind of the protrusions, the foldings on the surface of the brain. The sulci or a sulcus is just a very deep groove. So fissures, sulci are the shallow grooves. Fissures are the deeper grooves. So the sulcus, you can see, is a slight groove, but not as deep as that fissure. So it's the fissures that are going to separate the different lobes of the brain. Right? So like the longitudinal fissure is this very deep, deep groove separating the two hemispheres. Whereas the transverse cerebral fissure is the deep groove between the cerebrum and the cerebellum. So again, 5 lobes of the brain we'll look at. So the frontal lobe, and all of these are separated by sulci and fissures. Temporal lobe, right, on the lateral side of the brain, parietal lobe, occipital lobe, and the cerebellum. So you see some of these kind of follow the bone naming patterns of the skull. We looked at bones of the skull, so like the temporal bone, the frontal bone, parietal bones, occipital bones. So these lobes of the brain are also named in that same fashion. So several sulci divide these hemispheres into the 5 lobes. So the transverse cerebral fissure, we said, separates the main cerebrum from that cerebellum, separates all these other lobes. So the major sulci, or grooves that divide these different lobes, we said the central sulcus was kind of that primary one in the center of the brain region. So it's going to separate our precentral gyrus and frontal lobe from the postcentral gyrus of the parietal lobe. We also have a parieto-occipital sulcus, or a groove separating these 2 lobes, and a lateral sulcus outlining the temporal lobes. So each hemisphere has 3 basic regions. So we can look at the cerebral cortex, or the superficial outer layer of gray matter, the white matter, kind of filling the internal component of the central nervous system, and then the basal nuclei are these little pockets or islands of gray matter kind of deep within this white matter. So the cerebral cortex is kind of the executive suite of the brain. It's where your conscious mind, all of your thoughts, sensory perception, awareness, all of your voluntary motor movement, your memory, understanding, basically your personality is all in consciousness in this cerebral cortex region, just this thin outer layer. So it's composed primarily of neuron cell bodies and some dendrites, glial cells, but no real axons. And it only makes up about 40% of the mass. So again, it's a pretty thin outer layer of the brain itself. So this figure is just showing how different sensory and motor functions are kind of assigned to specific regions of the brain, sometimes called domains. So higher functions are spread over many areas. So when you see something, you're always going to use this region of the brain. When you hear something, right, that information goes to this side of the brain. So kind of this discrete cortical areas for different motor and sensory functions. So for general considerations to think about with the cerebral cortex, so just like the foundations of neural tissue, we talked about the 3 types of neurons. There are 3 types of functional areas in this cortex. So we have motor areas for our voluntary motor movement, sensory areas for your awareness of sensation, so like your vision, your hearing, taste, smell, right? And association areas. So how do we process that incoming sensory information? Also with the cerebral cortex, we have the contralateral function. So this just means that each hemisphere is concerned with the opposite side of the body. So left hemisphere is going to control the right side of the body, right hemisphere controls the left side of the body. So this lateralization or specialization of cortical functions generally only occurs in one hemisphere. But we use all of our senses, all of our consciousness in one way or another to involve the entire cortex. So the motor areas of the brain located primarily in the frontal lobe. So these are going to control our voluntary movement. So things that we have conscious control over. So the primary motor cortex is in this precentral gyrus. So remember, a gyrus is the ridge, the sulcus is this groove. So the precentral gyrus is just the ridge that's in front of or pre-sulcus. Right? Whereas when we look at the post-central gyrus, it's going to be behind or post the central sulcus. So the premotor cortex is just anterior to our precentral gyrus. So primary motor cortex is the darker red, premotor is this lighter red. region. Broca's area is a special area just anterior to the premotor area, but this is concerned with motor speech, right, with how we form language. Frontal eye field, also where we're going to control eye movements, the muscles that innervate the eyes. So our primary somatic motor cortex is located, we said, in the precentral gyrus, so that ridge just in front of the central sulcus. So this figure showing, so this is the front of the brain, the anterior, right? So the red is your motor map for the precentral gyrus, right? So we're in front of that groove. So there's large neurons allowing for conscious control of precise, skilled skeletal muscle movement. So we have pretty precise control over our voluntary muscle movements. So the premotor cortex helps to plan these movements. So it helps control either learned or repetitious or patterned motor skills. So actually walking, we have a lot of like muscle memory with this repetitive type movements. Also helps to coordinate simultaneous or sequential action. So it's just basically planning the movements out, which muscle needs to move at which time before we actually have to perform the action. So everything runs smoothly. We said the Broca's area is that motor speech area. So it's going to direct those muscles of speech production in the mouth, the tongue, the lips, the throat. So it helps to plan speech and direct those voluntary motor activities. Frontal eye field is going to control your voluntary eye movements. So we have 6 muscles attached to the eyeball that allow it to move in all directions. So the frontal eye field controls and innervates those eye muscles. So homeostatic imbalance, including damage to areas of the primary motor cortex, such as stroke. We can generally tell which side of the brain is affected by the stroke looking at which side of the body is impacted. So if the patient is paralyzed on the right side of the body, then we know the stroke had to occur in the right side of the brain. So again, left brain injury, left side of the brain is going to impact the right side of the body. So the paralysis occurs on the opposite side of the body where the damage occurred. The sensory areas are places of the cortex concerned with or conscious awareness of sensation. So it occurs in the parietal lobe, some in the insular lobe. Insular lobe is kind of an embedded, buried lobe inside, and the occipital lobes in the posterior region. So 8 main areas that we'll look at include our primary somatosensory cortex in this postcentral gyrus, somatosensory association cortex in the lighter blue, cortex visual areas here in the occipital lobe, auditory areas would be where you have your sense of hearing. Vestibular cortex would be your sense of balance. Olfactory cortex, gustatory cortex, and the visceral sensory area for your internal organs. So the primary somatosensory cortex, we said, was located in that post-central gyrus. So the blue is just behind that central sulcus. So it's going to receive general sensory information from the skin and stretch receptors or proprioceptors within skeletal muscles, joints, and tendons. So this also helps to allow spatial discrimination. So we're able to identify a specific body region that is being stimulated and receiving these sensory signals. So for instance, if a mosquito lands on the back of your hand, you're able to kind of discern without looking, just by your sense of touch, if it's closer to your knuckles or is it closer to your wrist. So we're able to distinguish between different locations of touch. Somatosensory association cortex, again shown here in the light blue, it's just posterior to the primary cortex. So again, association just means where we're going to integrate and interpret our incoming sensory information. So we're able to understand what it is that we're receiving, what we're looking at, what we're touching, what we're hearing. So it helps determine size, texture, relationship of the parts of the objects being felt. So a good example of this, ladies, is when you're digging in your purse for something, and you can't look in your purse because you're driving or whatever, but you're feeling for the object you're looking for. So once you find it, you can tell that that's what it is just based on touching it. Visual areas are going to receive the visual information from the retinas in the eye. So this is the primary visual cortex, just a very small region. So this is only straight incoming information from the eyes and the retina. Whereas the visual association area takes up much more room of the brain. So it's much harder job to process all of this incoming sensory information. So this visual association area uses our past visual experiences to help associate or interpret what it is that we're seeing. Right? So color, form, movement. But overall, these complex processing is going to involve almost the entire posterior half of the hemisphere. So we're going to need to call in all of our sensory association areas. Auditory areas, as in with the primary auditory cortex here on near this temporal lobe, superior margin of the temporal lobes, is going to interpret incoming sound information from the inner ear. So we can detect differences in pitch, loudness, or even location. The auditory association area is going to allow us to interpret what those sounds mean. Right? So we can perceive these sounds and understand them. The vestibular cortex is just in the posterior part of the insular lobe. So again, the insular lobe is kind of just embedded or buried underneath these other lobes. But is responsible for our awareness of balance or the position of our head in space. So your vestibular cortex, whenever you see vestibular, it has to do with your sense of balance. Olfactory cortex, of course, with your conscious awareness of odors. Right? So olfactory here of the temporal lobe is going to be involved with your sense of smell. Gustatory cortex is involved with taste. It's also found in insula, just deep to this temporal lobe. Visceral sensory area is also going to be posterior just to that gustatory cortex in the insular lobe. But this allows us to perceive sensations from our abdominal organs. So you're able to tell when you have an upset stomach or if you have a full bladder. Multimodal association areas, here shown in the kind of light purple shading, you see take up a large percentage of the brain space. So they're going to receive input from multiple sensory areas and also send outputs to multiple areas, both motor and cognitive. So this allows us to give meaning to the information we're receiving. We can tie it to previous experience. We can learn new things. We can then decide how to react to incoming stimulus. So pretty much all of your sensations, your thoughts, emotions, your consciousness, what makes you who you are, your collective experiences in life, are all going to be processed and stored in this frontal lobe area and used by these multimodal association areas. So broadly, we can divide these into three parts. So the anterior association area is just in this frontal lobe. Posterior association area, and the limbic association area. So your anterior association area, sometimes called the prefrontal cortex, is probably the most complicated region of the brain. So it's involved with intellect, cognition, recall, personality. It's pretty much who you are, your whole personality, you as a person, is stored just in this frontal cortex, prefrontal cortex of the brain. It also contains your working memory. So for abstract ideas, judgment, reasoning, planning, right, you can tap into your memory stores. So development of the prefrontal cortex also depends on feedback from social environment. So you have to kind of build that brain muscle. So 9 functions of the prefrontal cortex: empathy, insight, response, emotions, body regulation, morality. It's pretty much all of personality traits. We'll also look at the limbic brain, which is said to be kind of your emotional part of the brain. So it controls things like your fight or flight response. And a lot of your emotions live in this region of the brain. Posterior association area, just this large region in temporal, parietal, and occipital lobes, helps to play a role with recognizing patterns and faces, as well as localizing us in space. So some tasks like understanding written and spoken language would fall under the role of the Wernicke's area, right, within this posterior association. So we said the Broca's area was for your motor speech. The Wernicke's area is then how you understand and interpret written speech and spoken language. The limbic association area, part of the limbic system, involves cingulate gyrus, the hippocampal gyrus, and the hippocampus. But basically, this is the emotional portion of the brain. So it also plays a role in memory. So emotional impacts have a role in kind of transferring memories to your long-term storage. So kind of the higher your emotion during an event, the more likely you are to remember it. So a classic example is like 9/11. If you're old enough to remember when 9/11 happened, pretty much everybody remembers exactly where they were and what they were doing when 9/11 happened. So it has that large emotional impact, kind of those flashbulb memories. Another interesting thing about the limbic system, the olfactory bulb. So it's not an actual part of the limbic system, but it does kind of overlap. So there is a little bit of crossover with the limbic system and the olfactory bulb. So this is one reason why smell is one of the strongest triggers of memories. So lateralization of cortical function, we said that each hemisphere is separate, kind of has its own roles to play. So this division of labor between the hemispheres is called lateralization. Cerebral dominance is referring to which hemisphere is the dominant for language. Right? So are you left-brained person or a right-brained person? 90% of humans are left side dominant, meaning that they are right-handed. So remember that contralateral function. So the left side of the brain controls the right side of the body. So left dominance results in right-handedness. However, in about 10% of the population, the roles are reversed. So this results in a left-handed person. So there would be right brain dominant. So we've noticed some lateralization of different functions and roles between the hemispheres. So left hemisphere is more for logical task, math, language, whereas right hemisphere is more for kind of artistic pursuits. So visual spatial skills, intuition, emotional, artistic, musical skills, primarily found in the right hemisphere.